All right. Good afternoon, everyone. So today we are all delighted to welcome Dr. Jens Schmidt from Michigan State University for today's biochemistry seminar. So a brief introduction for Dr. Schmidt. He did his PhD in MIT with Dr. Ian Cheeseman studying kinetical complexes. I think he was using molecular biology and he learned some single molecule biophysics over there. And then he did his postdoctoral work with Dr. Thomas Check at Colorado Boulder, working on telomerase recruitment. And in 2018, he started his own lab at Michigan State University, studying genome maintenance using single molecule live cell imaging. And today he's going to give a talk on single molecule cell biology about genome maintenance. So let's welcome Dr. Schmidt for today's talk. Thank you.
Thank you, CJ. Can everybody hear me okay? Should I check with the Zoom people if they can hear? Are they good? Okay, cool. Okay, thank you for the introduction and thank you for the invitation to come talk about our work. It's always fun to come on seminar visits, especially when the sun is actually shining outside and it's not just gray the whole time, even though you're in meeting rooms most of the day, but it's been a great day. So what I want to convince you of today is that we can learn a lot of things from looking at single molecules rather than average populations of cells. And that one of the ways I like to illustrate this right off the get-go in my presentation is these two images, nondescript images of two cells with a fluorescent protein in the nucleus of a HeLa cell here, or actually a U2S cell. And you can't really tell them apart with this from this image. They basically look the same. But if you look at them at the single molecule level, what you'll notice is that the proteins on the left side are barely moving around, and the ones on the right are wiggling around really fast. So basically, the ability to detect single molecules can reveal things about the dynamics of a system that you can't otherwise visualize. And the way we do this in most of our experiments that use adherent cancer cell lines is we engineer a halo tag into the endogenous loci of the proteins that we want to study. And the halo tag is a small, like 35 kilodalton protein that can covalently interact with a ligand that we can append to any fluorophore that we choose. And then we combine that with high-low microscopy, where we use a turf microscope to shine a laser beam through the cell at a shallow angle to give us some optical sectioning and make sure that we can actually get the signal to noise that we need to detect single molecules. So that's how we do that in practical terms. And then in terms of what we can learn from these kinds of movies is we can learn about biochemical properties of the molecules that we're studying, but in the in vivo context in the living cell. So for example, we can learn about complex formation by analyzing how fast these molecules wiggle around in cells. So you can imagine a very large macromolecular complex is going to diffuse slower through the cell than a small protein, for example, just the halo tag by itself. You can look at associations with chromatin in the case of DNA repair factors, or sort of non-specific chromatin binding. But we can also then manipulate the system, for example, by inducing DNA double-stranded breaks, and then look how DNA repair factors respond to that, right? So how they change their dynamic diffusion and bind to the substrate in living cells. So we can basically make a lot of biochemical measurements without disrupting the cell by using this technique. And so in my lab, we have basically three different projects, two of which I'll touch on today. We study telomeres and telomerase biology, DNA double-strand break repair, and we also study autophagy, which is a separate project that I won't have time to get into today. So just a little bit of background about telomeres. So human chromosomes are linear, and they have repetitive sequences at their 3' end. Let's see if oops, my cursor's going to float over here. So they have repetitive sequences at their 3' end. Most of that repetitive sequence is double-stranded, and then there's a short single-stranded overhang. And basically what these ends look like to the cell is a double-strand break. So as you all are probably very familiar with, and CJ studies in his lab, is there's a sheltering complex that protects these ends and hides them away from the DNA damage response. So that is sort of the one problem that telomeres face, is that they look like a double-strand break, and we want to make sure that they're not getting repaired by the DNA repair machinery, or DNA damage repair machinery. The other problems that cells face is that when the chromosomes replicate, the very end of the chromosome can't be copied properly. So that manifests slightly differently on the two strands. So on the lagging strand, the synthesis of the RNA primer that's required for DNA polymerase makes it so that you can't actually copy the very end of the chromosome. And on the leading strand, you can copy all the way to the end, but you need a single-stranded overhang for sheltrin to carry out its end protection properties. So you have resection, and that regenerates a single-strand overhang, but also shortens the chromosome end. So in most cells, telomerase, in germ cells and stem cells, telomerase can elongate these chromosome ends to compensate for the DNA that's lost during DNA replication. But there's sort of a fine balance in terms of telomerase activity. So too much telomerase activity or telomerase upregulation in the inappropriate setting is a hallmark of cancer. So about 85% of cancers turn on telomerase to maintain their telomeres. And on the flip side, if you don't have enough telomerase activity because of mutations in the pathway that lead to defects in telomerase activity, you have premature aging diseases such as dyskeratosis congenita and aplastic anemia. So what we want to do in my lab is we want to really quantitatively describe telomere maintenance in human cells. And so in the past, what we've done is we've looked at telomerase recruitment to telomeres while I was a postdoc. So we analyzed how the telomerase here in green binds to the small dots that are telomeres in red in this movie that I'm showing here. And we've also developed a methodology to look at telomerase catalysis using optical tweezers in collaboration with Dr. Matt Comstock at Michigan State, who's an expert in developing these optical tweezer tools. And then what I'm going to talk to you about today is sort of the third important step in telomerase function, is its biogenesis. So, and what you can already appreciate from this little cartoon that I'm showing here is that there's two locations in the nucleus other than telomeres that telomerase or the telomerase RNA without its protein component show up in, and that's the nucleolus and the Cajal body. So I just want to really briefly touch on what nucleoli are. So nucleoli are these large phase-separated compartments in nuclei that are built on the ribosomal, the chromatin that's coding for ribosomal RNA. So there's ribosomal RNA that's made in it. The ribosomal RNA is processed, gets modified, is cleaved, and then matures and assembles with ribosomal proteins. And so there's small subcompartments of the nucleolus, but for the purposes of this talk, all you have to remember is that it's sort of a subcompartment of the nucleolus that has different biophysical properties than the general nucleoplasm, right? So in the talk, I'm going to distinguish a lot between the nucleoplasm and nucleoli. So what else you need to know about the telomerase RNA is that the telomerase RNA resembles a small Cajal body RNA. So in our cells, we have small Cajal body RNAs and small nucleolar RNAs that localize to those subcompartments respectively. They have a lot in common, but there's one small difference, is that's the association of this TCAP1 protein up here in magenta. So what these SNO RNAs and SCA RNAs do is they are responsible for pseudouridylation and O2 methylation of RNA bases in ribosomal RNAs and spliceosomal RNAs. And that's directed by discarin or fibrillary, which is associated with these RNAs. And so what's really interesting about this is if you look at this complex, the difference between this protein binding to this stem loop here, to this cap box in this stem loop, makes the difference of this RNA going to the Cajal body versus going to the nucleolus. And basically what the telomerase RNA has done is at its 3' end, it has this HACAA domain, which resembles a SCA RNA. And as far as we know, the main function of that has been to protect the RNA from exonucleolytic degradation. So if you get rid of the core component of this complex, which is the discarin protein, you get exonucleolytic degradation, and you reduce telomerase levels, which can lead to premature aging diseases. So I already touched on the cat box here. So this is from the cryo-EM structure of the telomerase holoenzyme. So you have basically two functional lobes. You have the HACA lobe up here, which I just showed in the previous slide, which associates with TCAB1, which unfortunately I color-coded differently here. And then you have the catalytic lobe, where you have the template region and the reverse transcriptase subunits of TERT, which actually carry out the telomere elongation. Okay, so the central player of this story is TCAB1 and how it functions in telomerase assembly. So what we knew from other people's work is that if you knock out TCAB1, telomeres shorten. So you get a reduction in telomerase activity. So this is apparent in this gel here. It's a southern blot of the telomere restriction fragments that get shorter and shorter over time if you don't have TCAB1. The other thing that we knew was that if you knock out TCAB1, the telomerase RNA, instead of localizing to Cajal bodies, which are these small bright dots in the nucleus, this is an RNA fish experiment, the telomerase RNA now, instead of that, localizes to nucleoli, which are these much bigger, more diffuse areas, which are also marked by discarin. So we knew that. So you get rid of TCAB1, the telomerase RNA ends up in the nucleolus. So with what we had noticed in our single molecule studies of the TERT protein here in green is, so we have telomeres in red and then a nucleolar marker in blue, is that if you look at this, these are all single molecules, each green dot, is what you'll notice is the green dots never diffuse into the nucleolus. And actually frequently they sort of get stuck at the edge here, like this guy right here. So TERT never enters the nucleolus, and the telomerase RNA ends up in the nucleolus when you get rid of TCAB1. So we had a very simple hypothesis that telomerase does not assemble in TCAB1 knockout cells. And so this work was done by Basma, Gloria, and Kate in the lab. Basma has since graduated, and it was really a big teamwork that involved everybody. So it's been a very fun project. So the first thing that we wanted to test is we can take these cells and we can, here's just a Western blot of the purification. So we can take our cells, we can immunopurify the TERT protein using an antibody, and then we can see what's stuck to it. So what is the assembly state of telomerase? And the first thing that we noticed is that when you do that, so here's the protein analysis by Western blot, here's the RNA analysis by Northern blot, what you can immediately appreciate in two different cell lines, either parental HeLa cells or cells that express a tagged version of the TERT protein, is that in the wild type cells, UIP a lot of telomerase RNA, and then knockout cells, UIP very little telomerase RNA, consistent with the idea that telomerase assembly is reduced under these circumstances. Okay, so then we also did this in a slightly different way, is where we, instead of, so the previous experiment that I showed is taking endogenous telomerase and immunopurifying it. In this case, we overexpressed the telomerase RNA and protein and did the same experiment. So again, you can appreciate that in the telomerase and the TKO, which signifies here that telomerase, TCAB1 knockout, we get less RNA, but we can then also use discarin as a secondary marker because discarin associates with the telomerase RNA, and so that in this case is also strongly reduced. So that's all consistent with a reduction in telomerase assembly when TCAB1 is absent. And then what we wanted to know is, how does that actually impact telomerase activity? So we can take our IP'd material and do a telomerase assay on it by giving it a short oligonucleotide primer and radioactive nucleotides, and it gets elongated in these beautiful 6 nucleotide patterns. And what you can see is that telomerase activity is also reduced. But when you're a good biochemist and you normalize to the amount of input enzyme by normalizing it to the amount of RNA that's added to the sample, you find that activity or specific activity is not reduced. So it's a pure reduction in telomerase assembly. Per molecule, the activity is comparable in the absence of TCAB1. Okay, so this is all very nice biochemistry, but I told you we wanted to look at this in living cells using single molecule approaches. So what we did was, what we learned previously is that because telomerase is a very complex RNP, we get different diffusive states of this molecule in living cells. So here's an example of this. So we can take these kinds of movies, we can do particle tracking in this, and then analyze the trajectories with a variety of methods. And what we find is that there's 3 different states of telomerase. You can see sort of molecules that are very static, that aren't moving around. Of course, at the end of the movie, they don't happen to appear that much because they bleach. But so there's a slow diffusing state or a static state, which is telomerase that's either associated with Ca bodies or telomeres. There's a slowly diffusing state, and there's a rapidly diffusing state. And the way we annotated these was to assume that the fast diffusing state was TERT alone. The slowly diffusing state was the whole big telomerase RNP diffusing through the cell. And then once it binds to something, it becomes static because these telomeres or Ca bodies don't move around very much on the time scale that we're imaging. So we can then also do those experiments with TCAP knockout cells and, importantly, telomerase RNA knockout cells as a control. And what you can maybe appreciate visually is that in the right 2 movies, these molecules move around quite a bit faster. But we can, of course, quantify all these things. And the first thing that we wanted to do is, okay, is this state assignment reasonable at all? So what we do is we can basically add together the fraction of molecules that move slowly and are static. So those are the molecules that we assume are assembled. So if we add those fractions together, it accounts for about 45% of the molecules. If we knock out the telomerase RNA, so these 2 states no longer exist technically, this drops to about 20%, which is sort of the baseline that we see with most proteins. So even if you have a molecule, if you just express, for example, the halo tag in the nucleus, you always get a static fraction about 15 to 20%. So it might just reflect the stickiness of the tag itself. And then when we do a TCAP knockout, we get a partial loss of this static fraction. Of this assembled fraction, right? So basically, we see about a 50% reduction of the number of assembled molecules by analyzing the diffusion patterns of TERT in this case, which is consistent with the pull downs, because also in the pull downs we saw about 30 to 40% assembly relative to the control cells. Okay, so this is one way of looking at this in intact cells, but we also wanted to use a different technique. This is now not an imaging technique, but we wanted to analyze the assembly of the telomerase RNP using RNP-map, which is a high-throughput sequencing-based technique. And aficionados might have noticed that Basma now changed her name to Klump because she got married halfway through the project. So the way this approach works is we use a bivalent cross-linker that reacts with lysine residues on the protein side. And then when we UV cross-link, after that, the UV cross-link is formed with the RNA. So it basically probes for, it will cross-link residues, lysine residues to RNA that's in close proximity. Then what we can do is we can play a fun trick that happens in a standard biochemistry trisol extraction, right? You have an aqueous phase and an organic phase. These cross-linked species will sit right at the border between those. So what we can do is we can basically separate cross-linked RNA from non-cross-linked RNA. And the non-cross-linked RNAs are negative control, right? So we can compare those 2 species. So we take that RNA, we digest it with a proteinase to generate small adducts. And then we use low fidelity reverse transcription that will read through these RNA molecules and incorporate mutations at the sites of protein RNA crosslinks. And then we can do high throughput sequencing and map those contacts then back to the RNA structure. Of course, as I just explained, we compare the mutational frequency in the crosslink versus the non-crosslinked sample to see which sites are significant. Okay, so the first thing that we wanted to look at is the TCAP interaction with the telomerase RNA, right? So here's this map or the structure from the cryo-EM map, And you can see here a couple of highlighted examples where lysines in blue are close to these respective bases in the RNA. And when we do this RNP map technique, here's basically color-coded the p-value of the significant hits that we get. Those two bases show up extremely nicely. There's significant hits in our analysis. And then when we knock out TCAP1, this one goes away and this one shifts. And that might reflect now that this 411 position might be able to now react potentially with a different protein, or it shifts slightly the profile. So that was a really, really nice control here, or a nice way to show that this technique works, right? So we can detect using this technique whether a protein binds or not. On the flip side, if we look at this region over here, again, lysines highlighted in blue, bases highlighted in red. This is where discarin binds, which should be unchanged in both of these genotypes. And we see that reactivity almost exactly matches between the two samples. So we can use this technique as a way to see what that TCAP1 binding is basically missing, but other protein associations stay the same. Now, what about the telomerase, the TERT protein, right? So we wanted to see, again, in living cells, in a TCAP1 knockout, is the amount of TERT associated with the RNA reduced or changed? And we can see here's just a couple of examples again, where we can see that there is differences in the pattern. And this is not a yes/no answer like it was for the TCAP1, because we still know in the case of the TCAP knockout that there is some assembly of the TERT protein with the RNA. It's just not quite as much as in the control cells. Okay, so I hope I convince you with these three different techniques. We've shown all the same thing. In a TCAP knockout, there's less assembly of the telomerase RNA with the TERT protein. So the question is why? What is happening in those cells? And so the first thing that we looked at was what I already showed you in the original movie, that had that TERT was excluded from the nucleolus. So we repeated that experiment in TCAP knockout cells. And what you can appreciate that is that the majority, you can see some molecules that associate with the nucleolus, but for the most part, this is quantified down here, most of the molecules zip around to nucleoplasm, right? So again, most of the TERT molecules are in the nucleoplasm. The RNA is in the nucleolus. So we just think that the spatial separation is inconsistent with assembly. So the question is the telomerase RNA trapped in the nucleolus and doesn't get out, or does it shuttle in and out? What's the mechanism of this trapping of the RNA in the nucleolus? So what we turned to is we wanted to look at this in living cells. And so we incorporated 3 MS2 stem loops at the 5' end of the telomerase RNA. And these MS2 stem loops can bind to the MS2 code protein, and that is fused to GFP to visualize these RNAs in living cells. And we integrated those stem loops into the genomic endogenous genomic locus. And what you can appreciate here, there's some really nice clones where we have exclusive expression of the tagged RNA, as you can appreciate by the size shift. And so then we took those cells and knocked out TCAP1. And as I previously showed with the fish experiments, you can see here in the control cells, you see these bright foci for the telomerase RNA, which correspond to CAHA bodies. And then for the RNA, you can see this accumulation of the RNA into the nucleolus. And what's even a little bit nicer about these images, what you might be able to appreciate is, if you look at the nucleolar signal here, there's bright areas and there's dim areas. And that corresponds to these different subcompartments of the nucleolus. And the telomerase RNA happens to co-localize with these dim areas, which is where discarin goes, which is where the post-translational modification of the ribosomal RNA is carried out. So then what we did is we took these cells and we took the cells and we did a fluorescence recovery after photobleaching experiment. So we can take one of these cells where we can see very clear localization of the telomerase RNA to the nucleolus, and we can zap it with a laser. and then we can watch as the signal recovers or doesn't recover in this case. So if there were active rapid shuttling of the RNA in and out of the nucleolus, we would expect this fluorescence signal to recover. But what we see is that there's basically no recovery or very little recovery of the RNA moving in and out of the nucleolus. So what we believe is going on in this case is that when you don't have TCAP1 bound to the telomerase RNA, it basically turns it into a snow RNA that now localizes to this dense fibrillar component of the nucleolus. And when TCAP1 binds to it, it prevents its recruitment to that site. So what happens in the cells then, when you don't have TCAP1, most of the RNA is stuck in the nucleolus. The TERT protein is outside of the nucleolus and effectively just can't assemble as much. Occasionally it happens, and you still have some telomerase assembly. That's why these telomeres shorten, but then are maintained at a shorter length. And so that was, and a lot of that, of course, is based on our studies of the single molecule dynamics of the TERT protein. So I'm going to take a brief break here. And are there any questions for the first half of the talk? Because I'm going to switch gears. Yes.
So you haven't mentioned the transcription step of the RNA. So
yeah.
It's transcribed outside the nucleolus or within?
That is a great question. We don't know. So that would be something that we want to look into, because it's important to learn whether it gets made in the nucleolus and then has to come out, or whether it's made outside of the nucleolus and then has to be prevented from getting recruited into the nucleolus.
So based on the genomic locus, would it be constrained to the nucleolus by being near the rDNA?
It is, as far as I know, not close to the rDNA, but we haven't carefully looked at this. But yeah, that would be a great experiment to do. Okay, so I will move on to a different project. So in this project, what we're looking at is how double-strand breaks are repaired in human cells. And so I don't think I need to convince anybody that double-strand breaks are really bad. And they can cause mutations, chromosomal rearrangements. And so they have to be repaired accurately and rapidly when they occur. And of course, there's a couple of examples like meiosis or class switch recombination, where double-strand breaks are purposely induced in the human genome to lead to crossover recombination for meiotic division, or for class, antibody diversity in case of class switch recombination. So the vast majority of double-stranded breaks are repaired by non-homologous end joining, which is the topic of this part of the talk. And I just want to introduce the players here for that. There is the Cu7080 heterodimer, which forms a ring that can thread on the end of a double-stranded break. There is the DNA PK, so DNA protein kinase catalytic subunit that, in combination with Cu, forms the DNA activated protein kinase. And then there is XRCC4, which forms a tight complex with ligase 4, which is that ligase that actually will ligate the ends together as part of repair. And then in the middle of the complex is XLF, which is an auxiliary factor. And so the way this is thought to work in human cells is that these proteins very rapidly associate with the end, and then 80 to 90% of the breaks, if they're blunt, are ligated back together very, very quickly and without any noticeable mutations. And of course, not every double-strand break in the cell is going to be a perfect blunt end that can be the substrate of this and just be put back together. There's also going to be cases where there's either adducts or short single-stranded overhangs that need to be processed before ligation can happen. And so there's been beautiful cryo-EM structures of these complexes where it first appears to form a long-range synaptic complex. Long range here signifies that the ends of the DNA are over 100 angstroms apart, so they're quite far apart. And in this short range complex, the ends are superimposed, and you can actually see the ligase being associated with the ends of the DNA that are getting ligated together. And it is unclear or the topic of many studies, at which stage this end processing can occur, and whether the DNA PK CS protein has to be removed for this step to occur, or whether it remains part of the complex. And so here's just a beautiful example of this. This is one of the structures. I just put in here, I talk about halo tagging, all these things. I like to put in here for like a scale comparison. Here's the halo tag, and here are all the proteins that we're tagging. So it's not insignificant in size, considering that DNA PKCS is 3,000 plus amino acid monster. So what we did is we tagged all of these proteins in U2OS cells with the halo tag. And here are just some nice gels to illustrate that these are all going to be cell lines that are homozygously edited, that are exclusively expressing the halo tagged allele. So DNA PK up here, Q70, XRCC4, and XLF. We tried tagging ligase 4 and it didn't quite work, but since it makes a very tight complex with XRCC4, we can kind of use XRCC4 as a surrogate for that. And just to mention this up front, all of this work was done by Maria Mikova, who is a truly outstanding graduate student in the lab. So we have all these cell lines. We wanted to make sure that they actually work, that the halo tag is not affecting the function of the protein. So what we can do is we can do clonogenic survival assays where we challenge the cells with zeaxanthin, which is a drug that induces double-strand breaks, and then look at the survival curves of these proteins and compare them to a DNA PK knockout, which is here in purple. And you can appreciate that there are some small differences, but for the most part, these cells can properly survive when they're challenged with double-strand breaks. So the first thing that we wanted to do is we took these cell lines and we can use the halo-tagged alleles in comparison with the wild-type protein to calculate how many molecules of these things do we have per cell. And it wasn't a mystery in the field. It was very well known that Q70 and Q80 are one of the most abundant proteins in the cell. So we have about 1.3 million copies of these per cell. So you can imagine they can very, very rapidly associate with a break when it occurs. From previous work, we knew that DNA PKCS was about 120,000 copies, so about an order of magnitude less DNA PKCS molecules. And then XLF and XRCC4 are even lower in abundance. So you can imagine that this difference in stoichiometry could have a big impact on how DNA and break repair is regulated. Okay, so how do people look at the recruitment of these proteins to breaks in living cells? Is they damage the cells and look for DNA damage-induced repair foci, which is here illustrated by another example, HaloRef1. So you treat cells with zeaxanthin, you get these beautiful repair factories where the breaks are repaired. And when you do that with all these NHEJ factors, you see nothing, which is expected because these repair factories contain many, many molecules. If you go back to the cartoon that I just showed, these breaks that occur and are repaired by NHEJ recruit maybe 2 of these molecules. So 2 of these molecules are by far not enough to give us a nice signal and to be able to analyze their recruitment to breaks in living cells. So of course, this is where our single molecule imaging comes into play. So we can take these cells, we can image them in control conditions here on the left, right? And you can appreciate they all zip around very nicely. And then when we treat the cells with zeaxanthin, we see many static molecules appear. One of the things that I didn't point out, actually, what was a little bit surprising, is that XLF is actually, for the most part, localized to the cytoplasm. And the nuclear concentration is not very high. So we believe that that's actually maybe one of the key factors that will control the overall rate of the repair here, is that the amount of XLF in the nucleus is limited. Nevertheless, you can see static XLF molecules in the nucleus of the cells after treatment with zeaxanthin as a break-inducing agent. Okay, so this is really cool, right? So now we can, something that was invisible by traditional microscopy approaches, we can now visualize using the single molecule method. And I don't know why I have to slide in here 4 times, but So what we do then with this data is we take our single molecule imaging movies, we track the particles, and we use the spot-on tool that was developed by Anders Hansen when he was in the TGen lab to get a fraction of the molecules that are associated with chromatin as a readout of the recruitment of these factors to DNA breaks. So that's just shown down here as a comparison. If you're using just the halo tag by itself, the fraction bound is extremely low because it just zips through the nucleus. If you fuse the halo tag to H2B, so it's going to be part of nucleosomes and then chromatin, the fraction bound is about 80%, so most of the molecules are static in this case. And then for all of the NHEJ factors that we tagged, when we treat with zeaxanthin, their static fraction is significantly increased. So that's just a proof of concept that we can use this approach to measure DNA break recruitment. Okay, so the experiment that we did then was we filmed every minute, we filmed a different cell, and we did that for a certain amount of time. Then we did a short pulse with the Colloquium, which, like zeaxanthin, induces double-strand breaks. It's just slightly more potent, so we're going to get a lot of breaks in this scenario. And then we kept filming cells for a period of about 60 minutes, and we looked at what does this fraction of chromatin-associated repair factors look like. And here's an example of that. So we can look at a whole bunch of cells as negative control before we induce damage. We induce damage here. It's a gray area because we're not imaging while we are inducing the damage. And then immediately after, you see an increase in the fraction bound that after a certain time, it goes back to 0, indicating that the breaks can actually be repaired, right? So it doesn't just go up and stay up because we're overwhelming the cells with a tremendous amount of DNA damage. We can actually repair this. Okay, so we did this with all of the proteins. So here again, this example was Q70. Then when we look at DNA PKCS, we see exactly the same thing. Immediately after repair, the fraction bound jumps up, stays high for a given amount of time, and then drops back down to the baseline. But importantly, it does so well before Q70. So there's about a 10-minute, so this takes about 30 minutes, the window width here, and DNA PK is about 18 minutes. So there's a long time span here at the end that where Q is still bound to chromatin, but the DNA PKCS protein is not. And like Q, XLF and XRCC4 stay all the way to the same time point, about 30 minutes after break induction. The other thing that you might notice here is that there's actually a delay for the recruitment of XRCC4 and XLF. It doesn't, like DNA PK and Q, the first cell you image after induction is already high. In XRCC4, especially, it takes a little bit, indicating that there has to be some processing or some events at the end of the break that delay its recruitment. And then we did the same thing by adding a DNA PK kinase inhibitor, and everything remains bound to the chromatin. So basically, if we are inhibiting the kinase activity of DNA PKCS, none of these breaks are repaired. So really showing that not only is DNA PKCS required as a scaffold to form this long-range complex, but it also is required for repair and the transition into the short-range complex where ligation can occur. So basically, I think what this data shows is it's basically evidence for the existence of these two different conformations of these repair complexes in living cells. But you might say, okay, hold on 1 second. We're filming a different cell every minute, right? So it's not the same exact breaks that we're looking at. So this is going to be a little bit complicated, and it's the first time I'm presenting it like this, so bear with me. So basically, if we look at the cell, we have all these NHEJ factors, and they're all present at different abundances. So, and I try to get the abundance as close as possible in the picture as a true abundance, but it's not, can't draw that many Q molecules because there's just so many. So now imagine what happens when we bind a break. Basically, we have enough Q to bind all of the breaks, but we only have enough of the other factors to actually repair one of them. right, as a hypothetical here. So then what happens when repair happens is that one gets repaired, and now all these proteins move on to the next one. So experimentally, what that should look like is from here to here, Ku gets released. So 2 molecules of Ku get released, but DNA-PK CS remains bound, right? Because it's basically operating at Vmax conditions where you have so much more substrate than it can handle. So it just jumps from one breaks to the next, and then it remains high. A very simple prediction from that is that the fraction bound of Ku should vary with the number of breaks, right? So if we make more breaks, the fraction bound of Ku should go up even more. So that's exactly what we tested. So we did a titration. We did the same exact experiment, but now we titrated in the amount of the Colloquium drug to generate more and more and more breaks. And so as you can see here, now this jumps up. Basically, the fraction bound goes up and up almost to 80% at the highest concentration. And this highest concentration is really not great for the cells. They don't seem to be able to repair everything. They don't look happy the next day. So this is a tremendous amount of breaks, but you can appreciate that as predicted from the model that I just showed, the fraction bound goes up and up and up. In the case of DNA-PK, that is the case for a certain range, but then instead of going up further, this plateau where we're in this scenario where DNA-PK is operating as fast as it can and is jumping from break to break to break to break, that plateau remains the same height, but just gets longer, right? So that's the window where we have more breaks induced than DNA-PK is ready to handle. So if you have a low concentration, see it goes up, and you can almost imagine this as a dissociation kinetics kind of an experiment. If you are saturating it, it stays on for a long time because now it has to jump from break to break. So this is a really, really nice way of showing that basically what the rate limiting factors for NHEJ are, right? So we have way more Ku. It can bind all the ends and make sure there's no funny business that happens at the ends that might limit the ability of NHEJ to ligate them back together. But then there's other factors that have to be recruited after that to complete the repair. So what we're doing now to follow this up, and this is just preliminary, so I don't have any data to show quite yet, is that we're using the same, a similar approach, but instead of imaging one cell every minute, we try to look at a single cell and image it for an extended period of time. And instead of imaging it as fast as we can, like we do for all the other single molecule experiments, we just take one image every second to map out exactly how long do these individual DNA repair factors actually stay bound. So what is sort of the real residence time of these molecules when breaks are repaired? And the cool thing is we can do the same trick. We can titrate the amount of Colloquium that we use to get either into a regime where basically the breaks are all repaired in synchrony, or we can put it into a regime where there's so many breaks that are going to be bound by Ku, but there's not enough DNA-PK-CS to repair them all at the same time. So here's just an example of that. Here's an untreated cell. And this now might look fairly similar, but it's a very different experiment because we're only taking an image every second. So all the stuff in the background is kind of getting blurred away. And the static molecules really stand out if they don't move. And so then we can induce damage with either 20 nanomolar Colloquium, and you can see some more static events that we can track. And then if you do 80, you see really a lot more of these long-lasting Ku binding events. And you can see now here the time scale, right? The time scale of the previous movies that I showed, the whole movie was probably 10 seconds. And now we're imaging these for minutes and minutes. And so that's what we're doing now with all these factors to really map out exactly what the kinetics are under different break regimes. And we're hoping to find out exactly what the rate limiting factors are and sort of what is the amount of breaks that a cell can repair simultaneously. So just to reiterate that here, what we're thinking is happening in this case of this particular experiment is that we get consecutive repair of these Ku-bound DNA breaks, and we can use this technique to map out exactly the kinetics of the transition of the long range to the short range complex and map out how long they spend in each of these conformations. So with that, I'm going to end and just want to highlight again the people that were involved in this work. This is Maria right here, who did all the work on the NHEJ project. This is Gloria and Basma, who did the work on the TCAP1 project. Kate had already left. She's now a graduate student at UCSF. And Maddie was also involved in the RNP map of the telomerase RNP. And with that, I'll end. Oh, and also, importantly, the RNP map would not have been possible without collaborating with Chase Weidman at the University of Michigan, who is the inventor of this technique. Thank you.
Questions for Jens?
Thank you for the talk. Sorry, I have a question about the first part of your talk.
Yeah.
So you showed data using a bivalent crosslinker to establish RNP interactions. I was just wondering, what was the scaffold that you were using for the RNA targeting portion of your bivalent?
Oh, it's a diazarine.
Okay.
Yeah. So it's a UV activated generates a radical, and from what I understand is that it can basically react with almost any part of the RNA, backbone, base. It's not like a DMS where it's like Watson-Crick specific. So it can't. I think it's agnostic to the pairing. But if you look at the, let me pull this up real quick. This is right here. Here. Of course, it doesn't have the looping in here. So if you look at these, a lot of times these pattern where you have the base pairing indicated in gray, it appears to me just sort of by staring at the data, a lot of times it seems like the base pair regions are not quite as reactive. I suspect that a backbone crosslink might not allow RT read-through, while a base modification might. And then because we PCR the whole RNA, I think we might not be able to see backbone crosslinks. That's why base paired regions are excluded.
Thank you.
Hey, that was a great talk.
Thank you.
I have a question about the XLF. So there was a paper or two a couple of years ago. So you mentioned that thing about the cytoplasm.
Yeah.
And there's a paper or two a couple years ago that mentioned, or that talked about how XLF, if sort of there was too much damage, it was shut down, phosphorylated by AKT, then pushed to the cytoplasm, and then repair sort of stops. Basically, it's just telling the cell, you can't get through this.
Yeah.
And so I noticed, so I have one question, and then one thing I noticed. I noticed that with a DNA PK inhibitor, that XLF1, although it is still bound to chromatin, it looked like there was a general, so I think it was the
It's sloping down a little bit.
Yeah, it's sloping downward. So I want to know if that's something that you see often. And then the second question is have you looked yet at XLF with increasing concentrations of clindamycin?
That is happening like over these days right now. Yeah. Okay. So we're looking at that. That's a really good point. So, but I really like that model. I think it makes a lot of sense. Is to limit that. Basically, you would limit overall repair capacity by XLF. One of the things that doesn't quite make sense there is that XLF is not like, if you go to like the sensitivity data, if you get rid of XLF, it's not quite that sensitive. So there seems to be some redundancy with other factors like PAX or other proteins that could basically mediate this short range complex formation, or even the long range complex formation. But I think I really like the idea that XLF is sort of the rate limiting factor for the whole process, and that it can be regulated.
I think that's interesting. And I guess, I don't know, with clindamycin, I know that it binds the minor groove. I also know that it killed Alexander the Great.
Yes, it's very, very toxic. Yes.
It's powerful, but I don't know. So what I was thinking is that with XLF1, or with XLF perhaps because it's got its linking capacity. Maybe in that scenario it doesn't matter that much, and that's why you don't see it. At the other end, you might see when it might be predictive. So if it doesn't have a long dwell time, it might be predictive of whether the cells will survive.
Yes, exactly. So I think the thing that I really appreciate about the lower concentrations is that you can see the drop back down to 0, right? It really looks like there's, and the cells actually look fine the next day. So I think if you did a sensitivity assay, they probably would be okay at that concentration. But it's absolutely true that it's an extremely potent drug, and that's why we're using it in this experiment, so we can have that really short window of exposure. I was going to mention one other thing, but now that's slipped my mind. Maybe I'll remember in a second.
Questions for Jens? Actually, I have a question. So what happens if you overexpress DNA PK, if it's limiting to DNA repair?
It's a good question. I'm trying to think here. So my collaborator, Kathy Meek, she has a lot of very strong opinions about the expression levels of DNA PK and how they correlate with ATM expression and how that influences. So there's other factors that play a role here. I would assume that overexpression here of ATM wouldn't really change very much because the other factors are still at a lower level. It might accelerate repair, but it seems like there's a switch where DNA PKCase autophosphorylates itself, and that's what's critical for it falling off the DNA. So if you don't have that autophosphorylate, so the effect of the inhibitor here, we can mimic by mutating the autophosphorylation sites in DNA PKCase. So it's very critical, but I think based on the abundances, I don't think it would change the overall capacity very much because where we were just discussing XLF being sort of the limiting factor in terms of abundance also and nuclear localization.
Right. And then DNA PK is like a really big protein, right?
Yeah, it's over 3,000 amino acids. Yeah.
If no other questions, let's give Jens another round of applause.